Hello! On behalf of Metagenomic Ninjas, I'm super glad to welcome you all to our final presentation of our last and final stage for HackBio Internship 2021. Before we begin, let me introduce our team members and mentors. Our team included of 20 hardworking members who worked really diligently to achieve the successful results. Our team was Priyanka Singh, Isha Barve, Premnath, Suchitra, Shabnam, Lakshmi, Devika, Ifi, Mohammad, OJ, Sneha, Karthik, Ashatu, Rishikesh, Halak, Modli, Pratyuksha, Christabel, Akanksha, and Priyanka Mishra. This team was led by Priyanka Singh and Isha Barve. I, Isha Barve, would be presenting our work in front of you all today. We are really fortunate to work under the supervision of Dr. Sami Wambua, who is a professor in Pwani University, Kenya. Mr. Aidobu Olaboy, who is a PhD student in African Center of Excellence for Genomics of Infectious Diseases, Redeemers University, Nigeria, and Abdul Salam Toyin, who is a PhD student at School of Psychiatry, Faculty of Medicine in University of New South Wales, Sydney. Sir, it has been a pleasure working with you all. Moving on to our topic, let me introduce our title to you. A metagenomic approach to unravel the microbial diversity and detect the antimicrobial resistance genes present in the aquatic environment. Microorganisms are the essential component of the aquatic environment and are responsible for various biogeochemical processes. Their communities represent high aquatic biodiversity in the environment and comprise of bacteria, archaea, fungi, and viruses. The significant heterogeneity and microbial abundance in natural ecosystems is essential for their productivity and is critical to the sustainability of macro ecosystems at large. However, the microbial community in aquatic environment is dynamic and highly influenced by many external factors like antibiotics, their residue, and antimicrobial resistance genes. Presence of such antibiotics and their residue can impact the microbial diversity and their functions at different trophic levels. Aquatic environments have been labeled as genetic reactors or hotspots for AMR genes. AMR genes are places where extensive genetic exchange and recombination can occur, influencing future resistance profiles. In such a natural environment, the development of resistance within the microbial strains is considered to be a result of the concerted activities of the mobile genetic elements, which are fragments of DNA that encode resistant determinants and can move within or between DNA molecules, thus enabling the bacteria to resist antimicrobial chemotherapy. Excessive human contamination, industrial and hospital effluents are the major contributors of AMR development. These kinds of activities in aquatic systems have created selective pressure for the resistant microbes, leading to an increased prevalence of AMR gene in a variety of microorganisms. The ecological selection pressure will result in alteration in microbial abundance and diversity, which will ultimately affect the natural processes in the environment and will extend to the larger ecosystem. The impact of AMR in aquatic environments is extensive, threatening both humans and the environment. It is therefore of utmost necessity to detect the AMR gene reservoir and identify the microbial diversity within it through genetic analysis to effectively implement the control and prevention strategies for AMR dissemination. With the advent of metagenomics, the genetic analysis for characterizing microbial communities directly from the environment has been very convenient. The microbial diversity and functions can be greatly explored by adopting this method of analysis, and comprehension information can be extracted. The most common tasks in metagenomic analysis involve the taxonomic classification, functional analysis, metagenomic assembly, and comparison between metagenomes. Since we know that the complexity of antimicrobial resistance in aquatic environment can be unraveled by a detailed understanding of the genes at molecular level, this study uses metagenomics approach to explore the AMR gene and microbial diversity in the aquatic environment metagenome to unravel the comprehensive information. We had three objectives in mind while carrying out this project. 1. To explore the microbial diversity. 2. To investigate the presence of AMR genes and their functions. And 3. To compare and analyze the inferences obtained from two lake datasets. Hence, this study aims to highlight the importance of metagenomics to explore such antimicrobial resistance genes and microbial diversity in a given aquatic setting. In this study, we worked on two datasets from two places. First, Kazipalli Lake in Patancharu region, close to Hyderabad in India, which was polluted by direct dumping of waste from pharmaceutical production. Second, Nyadalason Lake, which was close to Umeå in Sweden. This was analyzed in a directly comparable way. The Sweden lake is a healthy lake with viable populations of fish, insects, and other fauna. It is used for recreational and swimming activities in summer, and it does not receive any direct discharges of sewage or industrial waste. Now let's get to our workflow. Our workflow consisted of three main steps, which were data acquisition and sequence analysis, taxonomic profiling, and detection of AMR genes. Here is a list of tools and their respective functions that we used for our analysis. Now I'm going to explain our method in detail. Data acquisition and sequence analysis. The raw sequence from open access database of ENA browser with the accession number PRJEB6102 was included in the study. The FASTQ file of Indian lake and Swedish lake dataset from the same project was downloaded for the analysis. The high throughput sequencing reads were paired end and were separated as forward reads and reverse read files. The paired end reads were analyzed separately for each lake. These files were uploaded in the Galaxy platform, and all analysis was done using the Galaxy platform. Quality assessment of raw reads was performed using FastQC. The minimum reads length of 30 base pair and the quality cutoff value of 28 was set while trimming the reads using CutAdapt. After trimming, reassessment of the read quality was confirmed using FastQC and MultiQC tool for the combined report of the quality assessment of both the reads. Since the MetaFLAN2 takes FASTA as an input file, the processed FastQ dataset file was converted to FASTA file using FastQ to FASTA converter. The output file generated was used as the input file for MetaFLAN2 analysis. The value of minimum total nucleotide length for the markers in a clade for estimating the abundance without considering the abundances was set as 2000. The parameters were set such that the profile of viral eukaryotic bacteria and archaea organisms was retrieved as the output. The quantile value for the robust average was assigned to be 0.1. Detection of AMR gene. De novo assembly was carried out on the processed dataset using the tool MegaHit. Since no pre-processing such as partitioning and normalization is required for this tool, the quality assessed data was directly used as input. The assembled genes in FASTA format were then scanned for antimicrobial resistance genes using Abricate. The default database used for this analysis was ResFinder. Sequence analysis. The quality assessment procedure trimmed the reads with a predefined parameter of about 30 base pairs in length and quality score of 28. 33,466,587 read pairs of Indian data and 69,474,306 read pairs of Swedish data was processed, out of which 9.2% and 12.2% was filtered out due to short length. The details of the trim reads in base pair is provided in the table which is present on the screen. This table gives us a summary of the quality of filtration of each read of Indian and Swedish dataset obtained using FastQC and CutAdapt tool. Further analysis was done using these filtered reads. Now let us move on to taxonomic analysis. Analysis was carried out using Krona pie chart. The taxonomic analysis of metagenome of the Indian lakes showed that 84% of the metagenome was predominant with the bacterial kingdom, followed by 16% archaea and 0.07% virus. On the contrary, the Swedish metagenome showed predominance of virus with 40% abundance, followed by 30% bacteria, 28% archaea, and 2% eukaryota. There was no unclassified or unassigned genes in both the metagenomes at the taxa level. Within the bacterial taxa, at the phylum level, the Indian metagenome had 49% proteobacteria, followed by 32% thermotogae, 11% bacteroids, 5% firmicutes, 2% spirochetes, and 0.3% chloroflexi, while the Swedish metagenome had 73% of cyanobacteria, followed by 19% proteobacteria, 5% actinobacteria, and 4% acidobacteria. A significant difference in relative abundance among the bacterial class between the two lakes was observed. The abundance details at the order level are given in figure 1A and 1B, which is displayed on the screen. The 5 most abundant bacterial genus in the Indian sample were Mesotoga, Desulphococcus, Idiomarina, Cangiella, and Pseudomonas. Likewise, in the Swedish dataset, the 5 most abundant bacterial genus were Pseudonabena, Anabena, Methylobacter, Synovium, and Thiomonas. Within the Archaea taxa, both lakes had archaea belonging to phylum Euarchaeota and class Methanomicrobia. But at the order level, Indian data had 98% methanosarcanales, 2% methanomicrobials, while Sweden data had 11% methanosarcanales and 89% of methanomicrobials. Within the eukaryota, Indian data did not have any eukaryota as per the taxonomic analysis. However, in Swedish data, 2% eukaryota which belong to phylum Apicomplexa was identified to be Hemiara. Within virus, Indian data had virus of Sarcoviridae family but could not be identified further to species level. Swedish data had about 22% of unknown viruses and 78% of Cardovirinae order. This 78% belonged to the Spiroviridae family of the Enterobacteria phage lambda. The AMR screening revealed the presence of 27 different types of resistance genes in Indian data, while 1 resistance gene was found in Swedish data. The resistant gene present in the Indian dataset confers resistance to major classes of antibiotics such as beta-lactams, aminoglycosides, quinolines, and marcolines, while the AMR gene of the Swedish dataset conferred resistance to aminoglycoside class. In Indian metagenomes, 7 AMR genes providing resistance to macrolides, 5 genes providing resistance to trimethoprim, 4 genes providing resistance to aminoglycosides, 3 genes providing resistance to beta-lactams, 2 genes providing resistance to sulfonamides, 1 gene providing resistance to chloramphenicol, and 1 gene providing resistance to rifampin was observed in the Indian dataset. The details of AMR gene and their resistance is given in table 2. To conclude, the microbial diversity between the two lake samples was distinctively different, suggesting a unique characteristic environment of each lake. Further, the prevalence of AMR genes suggested high selective pressure in the Indian lake and labeling the lake as a reservoir of AMR gene. The finding of this study indicates that the taxonomic distribution and AMR gene prevalence can be used to explore the characteristics of the environment itself using the metagenomic analysis as a tool. Given the broad range and relatively large abundance of known resistance genes we could identify in the Indian lake, it would be surprising not to find as yet undiscovered resistance genes in this environment. Many antibiotic resistance genes encountered in clinical settings are thought to originate from environmental bacteria where they have probably been present for millions of years. It would be astonishing if this gene flow suddenly stopped and the novel antibiotic substances we discover would not face horizontally transferred resistance development. However, the more likely scenario is that new resistance genes await us down the line. Large-scale screenings of heavily contaminated environments similar to the Indian lake could provide us with important insights into the range and nature of resistance genes that we might face in clinic in the future. The findings of this work and previous studies of the environments contaminated by pharmaceutical production waste suggest that much more effort is needed in unearthing risks related to pollution from production facilities. More studies are needed on resistance genes and their mobility in highly polluted areas as well as neighboring milieu. And further on in human microbiome of their inhabitants. It is of high importance to identify such pollution sources and to inform management. Furthermore, we also need to understand how resistant plasmids evolve and spread, and which factors can assist in such processes. Metagenomics can play a significant role in identifying such settings of particularly high risk and thereby suggest where to start future mitigation efforts. These are the references we refer to for making this PPT. We would like to express our gratitude to our team's mentors, Sami Wampua, Idovu Oloboe, Abdul Salam Toyin, who guided us throughout the project. We wish to extend our special thanks to our admins who were always there for us and helped us whenever we faced any kind of technical difficulties. We would like to thank our friends and families who supported us unconditionally. We would like to thank HackBio for giving us this wonderful opportunity in shape of an internship. Last but not the least, I would like to thank all our amazing viewers who listened to us patiently and encouraged us throughout the way. Thank you all once again. Thank you for your time.